So our next speaker is Runxian Zhang.
Zhang, yes.
Correct me.
Runxian Zhang, yes.
Okay. From the James Hutton Institute in the UK, and he's going to tell us about innovative advanced computational solutions for improved gene and transcript level analysis using RNA-seq.
First, I would like to thank the organizers to give me this opportunity to present our work. And there are several talks this morning already highlighted the importance of genome transcript annotation. And we're going to continue on that stream, but focused on the transcript quantification accuracy there. All right. So in the past few years, new alignment-free quantification methods like SAML, Callisto, they have been developed. So they're extremely fast. They're 20 or 50 times faster than the conventional tools, and they are also among the most the most accurate tools out there. They save time and storage, but the problem is that they do require a transcript reference. Just for those people who are not familiar with this, a transcript reference is almost like a dictionary if you want to find which word appeared in your book for how many times. And I work in a plant research institute, so all the data will be on plants. So Arabidopsis is about the best annotated plant species out there. But if you look at official annotation from the TER10 and Aeroport, we know there's a lot of transcripts and alternate splicing events are missing. So that will impact the quantification accuracy. All right, if we know that the reference is not good enough, why don't we just assemble the transcripts ourselves using the RNA-seq data we generated? The problem is, even for the cutting-edge transcript assemblers using short reads, you have about 50 to 80% of misassembled transcripts, according to different statistics. And also, like for the transcripts within your sample, from 50 to 80% of the transcripts are actually missing. So I'm just highlighting some of the problems with this transcript reference. You could have redundancy. For example, you have the transcript appearing multiple times, or you have transcripts that contain false splice junctions, or that transcripts actually only exist in fragment forms in your annotation. So we have actually started this work from 2014, and we generated high-resolution RT-PCR data just to validate the quantifications from the RNA-seq quantification. So I'm not going to go through this, but basically we went through at least 30 rounds of identified problems, proposing solutions, and tested solutions. And we come up with this AT-RTD quasi, which is a reference we put together for quantifications. I'm just going to show you the impacts of two filters we applied. Of course, there's a lot more details as well. So just by removing the transcripts with poorly supported splice junctions, we remove about 50% of the transcripts that put together by cufflinks, and 30% put together by a string tie. And also, the quantification has been strongly affected by the variations that coming from the misannotations at the 5' and 3' UTR regions. So we identified these problems in Arabidopsis. Actually, there's actually publications coming out this year have verified different aspects of these problems we identified. For example, from the Mark Robinson's group, they found the misannotation in the 3' and the human data also affects the quantification a lot. And from the Carl Kingsford group, they find the incompleteness of the reference transcriptome also cause problems. And similarly, you got duplicated context, that means you got redundancy. And family collapse context, that just means that you only find like one isoform out of several transcripts within a gene family. That will impact your transcription accuracy. Right, good. Later, good, that helps. So, as I mentioned, we have this high-resolution RT-PCR data. What you're looking at here is actually 762 data points over here. You have the RT-PCR quantification over here, and this is the quantification using Salmon, using the latest artificial rhabdopsis transcript annotation. You have Spearman and Pearson correlation from 0.5, 0.3. And using our reference, we are able to improve it to 0.8 and 0.9. And there's a lot more details. If you found that, if you want to find out more, you can go to this publication in AR. And this work has been published, been transferred into barley to get the reference for barley. And also, I got a PhD student working on potato reference. And we just got this project funded by the BBSRC that we're going to construct this kind of automated pipeline to get this transcript reference for all plant species and also beyond, because the methodology applies to human and other species as well. There's no reason that shouldn't work. So we are also recruiting a 3-year tenure track bioinformation. So anyone interested is welcome to contact me. So after you get the quantifications from the RNA-seq data, the next step is to do the differential expression. So we developed this tool called 3D RNA-seq, which carried out differential gene and transcript expression, and also differential alternative splicing and the differential transcript usage. So this is just show you 3 examples. Here you got differential expression at the gene and transcript level, but there's no change at the alternative splicing. And here you have changes at the alternate splicing level, but there's no change at the gene level. So in this case, you have changes at both ends. So this example just shows the importance of doing this differential alternate splicing analysis. So we have the red and green line that indicate the abundances of 2 transcripts within a gene. And the blue line is the sum of these 2. So that's the gene expression level. So in this case, you know, for some comparisons, you don't see differential expression at the gene level at all. But at the transcript level, you see massive changes. This will not be captured if you only do the differential expression at the gene level. So I'll just briefly tell you what's behind this 3D RNA-seq. We adopt the best practices using different PCAs, for example, to examine the data quality and also data normalization steps, etc. And then we use the lemma as the engine to do the differential expression. So this 3D RNA-seq app is, you can carry out the whole analysis without writing a single line of code. So it's very easy to use. It's a web-based graphical interface designed for biologists. Of course, I think it saves a lot of trouble for bioinformaticians as well. It has enhanced alternative splicing analysis, as I mentioned before, and it can cater for complex experimental designs. We say it's 3D analysis, but you probably spend most of your time actually doing the Salmon or Callisto quantification and to get data into format. You actually only need a few hours to analyze your data through 3D RNA-seq, even if as a beginner. And also, lemma has shown good control of false positives in literature and also in our studies. So for this 3D RNA-seq, for every step of the analysis, we have visualizations embedded within. So you can generate publication quality figures and tables from this analysis. I think the best thing is that at the end of it, a customized report will be generated that recording all the parameters you have been used in your analysis, including all the figures and tables and the references, explain the methods behind each step. So you basically have half of your publication ready. So this just shows some of the figures we have. And this is actually an ultimate guarantee of a consistent and reproducible analysis. Because with that report, if you provide the same input and same parameters, that's what you get. So just, we have this preprint on bioRxiv since June the 1st. We already have here about 900 downloads of the PDF files. And we have more than 500 users globally have tried out this tool. And we're going to carry out a training in Leeds as well in October through the Garnet, kind of plant community forum. So this just shows you that with the tools we developed, we're able to identify thousands of genes that only regulated at the alternative splicing level, which is a key component of this regulation. And here, you also see massive changes when the plant responds to cold stress at both transcriptional and alternative splicing level. So this is actually an important layer of regulation that should be examined. So we also developed this tool to identify isoform switches for time series data. So you can see you have isoform switches across the time series here. So this has been incorporated into the 3D RNA-seq as well. You can do that easily. Currently, we are developing tools to better translate the transcripts and characterize the functions of them. And this just shows you that using tools that we developed, we identify 900 isoform switches when the plant was transferred to cold stress. And 3/4 of them are actually protein coding. So just summary, with the tools we developed, including better transcript reference for RNA-seq quantification, and also this differential expression analysis tool, 3D RNA-seq, and TSIS, time series isoform switch, and accurate translations and transcript characterization. We're able to investigate alternate splicing in high resolution. So finally, I would like to acknowledge my collaborator, Professor John Brown, in University of Dundee, and also Professor Hugh in University of Glasgow. And Dr. Wenbin Guo as well. He's a postdoc who developed the 3D RNA-seq. Thank you. Okay.
So how do you evaluate your accuracy for all of those various tools? I'm very interested in splicing specifically.
You mean the RNA quantification?
Yeah, for splicing variations.
Yes. So we basically generate a high-resolution RT-PCR panel of about 62 genes. They're involving hundreds of transcripts, but we also have 3 biological replicates. So that gives us 700 data points on that plot. Go over here.
How do you know that your splicing quantifications is accurate?
So this alternate splicing panel has been established in Professor John's lab for several years. So we use, basically, you design primers with GFP on it, and then you kind of sequence that fragment. It has very high resolution. It can detect fragments that it's just one nucleotide difference. So yeah, so here is actually the ratios. We calculate for that specific event. We calculate the ratios of that event divided by the protein coding transcript. Then we have all these ratios over here. Then, of course, you can do the same with the RNA-seq quantification coming from salmon as well.
Mm-hmm. Okay.
So you basically just look at the correlations.
I see. Okay, yeah, we can take it more offline. Thank you. Okay. Let's thank Runxian again.
Great. Thank you.
So how can we do this quickly?